and specifically about not just the lymph nodes, right? That's part of it, but specific and general immunity. Now, part of general immunity or non-specific immunity is going to be physical barriers, lymphatic cells or T and B cells, and neutrophils and stuff like that, and then chemical mediators that helps out in the background. So we're going to have these classifications of non-specific resistance. The cells that are non-specific, those are the cells that we've already kind of talked about in chapter 19. Your neutrophils, your eosinophils, your basophils, your macrophages. All those class of cells will just swallow a cell up, a virus or whatever, kill it, destroy it. It doesn't matter what the virus is. The virus can be Ebola, can be anything, right? So what we're going to see is that we're going to have proteins, specific ones, that are polymorphic, that are found in lots of different virus classes or bacterial classes. And those proteins will then attach itself to receptors on the neutrophils. And those receptors are very non-specific. They're general. They will bind to these other non-specific proteins sticking out of the class of bacterium or the class of virus. So that is the big difference here, is that there's no specific protein that recognizes one specific epitope, right? That's what we're going to see in adaptive immunity, where we have a T cell that recognizes a specific epitope. An epitope is just a protein and sugar combinations that stick out of the virus, that sticks out of a specific bacterium. So we're going to see what makes E. coli E. coli. What makes these viruses slightly different. Now, we're going to have coronaviruses everywhere. We're going to have coronaviruses that's around us right now, but yet we don't have this kind of novel COVID-19 freak out that we see. So what happens is there is some kind of, because coronavirus is actually an RNA virus. So it is a, in order to have infect and then duplicate the DNA, you're going to need to have some kind of reverse transcriptase. So then you can make RNA that they have turn into DNA, and what we're going to see is they're going to cut their DNA, insert it into our machinery, our DNA, and then use our DNA polymerase. So then they can reproduce their genetic code. Then they'll use our messenger RNA, right? They'll use our RNA polymerase to make messenger RNA, which is their genetic code. So all of this is just happening, and they're using our own machinery to make copies of itself over and over and over and over. Now, besides the specific genome that is different between the COVID-19 and any of the coronaviruses we have right now, there's specific areas on the plasmid, the external shell of a virus, that will also be specific for that COVID-19. So it will have, in other words, protein combinations slightly different than the other coronaviruses that's always around us, because those protein arrangements are slightly different three-dimensionally. What happens is the receptors that we have that was saved up, the memory T cells, memory B cells that were saved up when we encountered coronavirus before, those receptors and those cells with those receptors, well, those receptors now don't recognize this new arrangement of proteins. Because the new arrangement of proteins is different, the receptors that we saved up, the T cells and the B cells with those receptors, what happens? Well, they don't recognize it. So it gets infected again. And you get more and more infections as a result of this. So this is why we having this, infection from this virus, even though we see a class of this virus previously. And previous infections with the coronavirus usually causes just mild illness, and that's about it. But you see something like MERS, something like SARS, those are also coronaviruses. SARS had a mortality rate of 30%. We believe that, I read at this new Medscape article, that the mortality rate of this new coronavirus is going to be less than 1%. When we have all the tests out there, right? And then we can do tests on all the infections we think are caused by coronavirus, we're going to see that there's a lot of people that actually has this infection. And very few of them are actually going to the hospital, right? And I believe they say like among the people who are sick, right, 24% has this endpoint result where you have people that are actually going to have to go to the hospital, that have to go and be intubated. So that's an endpoint result. And usually the people that get to that point, they get real sick, they're older. They already have a lot of pre-existing conditions. So far, we haven't seen the young kids get sick. That's a good news. Normally, like even the flu, do you guys know how many people die of the flu every year? 20,000 people die of the flu every year, influenza. 20,000. So when you hear about this freak out, yes, right, it can kill you. Will it? You're probably more likely to get into a car accident and die, right? Now, don't get into a car accident and die, all right? But yeah, that's probably more likely to happen. So what else do we see in terms of these? Now, I can go into detail, right? Those specific receptors that are found on neutrophils, on eosinophils, on natural killer cells, those are called toll-like receptors, TLRs. And those toll-like receptors recognize very general conformation of proteins found on classes of antibiotics. So you see like a cell, right, a bacterial the cell wall, a gram-positive kind of bacterium like strep and staph. Well, all of those have specific epitopes, making it strep versus staph. They also have very general proteins that's found on all gram-positive microbials, right? So those gram-positive microbes that have those very general proteins, those proteins are called PAMPs, P-A-M-Ps, right? And they're found through all the classes of those gram-positive, and then with gram-negatives, they share also similar proteins that our cells can recognize. Now, when we talk about the non-specific immunity, interferons are important. So there's no, when I say it's important, I mentioned it more than once, it's going to be on the test somehow, right? So interferons, and what makes it important is this. With interferons, interferons are now used as a way of combating viral infection. If you actually look and you are watching TV late at night, you will see commercials for people with hepatitis B, hepatitis C. Those people will then be given something called Copagus, which is basically a form of interferon. The interferon that we inject in you is called gamma interferon. It's stronger, and it's an additive agent to what our body already is producing. So what happens is now we're introducing more interferons into the bloodstream. It's going to go into the liver. Those interferons are going to hopefully prevent viral replication, cause neighboring cells to produce antiviral proteins. So then you can't have those proteins being produced, and you can't have the actual virus coming in. The weird thing is this. In order for a virus to even get into our cells, it needs a way of entering through the plasma membrane. And the way it usually happens with coronavirus is that it acts and it binds on a receptor, right? Those receptors that we mentioned for angiotensin receptors, right? ACE receptors, that's usually how coronaviruses enter our cells and infects the cells of the lungs and causes us to be sick. The weird thing is this new coronavirus doesn't enter that way. So we're trying to figure out how does it enter our cells if it doesn't enter this way. I can imagine that if we have because with hepatitis B and C, that become a chronic kind of infection. You can have that for months and months and years when you have hepatitis, and then it leads to cirrhosis, things like that. Well, if you have that kind of long-term infection, interferons and long-term use is actually pretty good. There's like a 85 to 90% cure rate if you're given interferon in time, right? Which is a pretty good cure rate, Usually we don't see that kind of high cure rate for any kind of infection. So when we have people with hepatitis B and C, and we give them interferon early enough, 85% cure rate. My dad actually got cured because of this. So that happened when I was in medical school. He having surgery, and I don't know what happened, but he got hepatitis from it, and hepatitis B. And back then, this was about 15, 20 years ago now, this was just very novel kind of a treatment, right? Now we see it a lot, this interferons, and this becomes our baseline treatment for chronic hepatitis. And then we talked about, as well, complement proteins. So know the difference in terms of alternative and classical pathways. Just very general, you know, which one is which one uses, which one allows us to activate or gets activated from our specific immunity, classical pathway, which one allows us to activate the innate non-specific immunity, the alternative pathway. Again, right, complement are a series of proteins, 20 different group of proteins, a group of 20 different proteins that when active will either bind to the virus or bacterium. As it binds to it in the alternative pathway, it's like a sign, it's like a flag. That flag then draws macrophages, draws more white blood cells to that site, right? Or once we've already activated the specific immunity, complement can also help our antibodies and help our immune cells. So with the classical pathway, antibodies bound to antigens activate complement, then they form membrane